Good morning, everyone. We're going to allow a few minutes here for everyone to collect and connect, and then we'll start. Okay, it's 9:32, so we'll start. Good morning and welcome to the Department of Neurology and Broad Stem Cell Research Center Zoominar on this Tuesday morning. My name is Tom Carmichael. I'm chair of the Department of Neurology at UCLA and interim director of the Broad Stem Cell Research Center. And it's a pleasure to welcome so many that are involved in the support and the help in establishing new directions in these two entities, the Department of Neurology and the Stem Cell Research Center. The Broad Stem Cell Research Center, as a bit of introduction, is a tremendous envelope that fosters scientific growth and development across many parts of UCLA. There are over 250 faculty in 55 academic departments, and it spans the School of Medicine, the School of Law, the School of Engineering, the College of Letters and Sciences. And so it really unites a lot of different disciplines and ideas under the guise of discovering stem cell biology and then moving those findings into the clinic. It has several major functions that do that. It funds excellent research cores that provide cutting-edge technologies to the Stem Cell Center faculty. It funds an innovation grant program, which is entirely dependent on philanthropy and really allows stem cell scientists and clinicians to do bold new things, to go new directions with innovation grants. And then it funds a fellowship program, and we'll hear from one of those fellows today, Dr. Samarasinghe, and this trains the next generation of stem cell biologists. The Department of Neurology, to switch gears, is one of the leading departments in the country in neurology. It's had one of the top NIH funding profiles for decades. It has the highest impact factor in its publications of any neurology department, so the most cited journals. And it has a large bench. There's 130 faculty in neurology that span the basic discovery science to clinical research and clinical trials to move therapies to the clinic, and of course a deep clinical care component. And so a final thought is, what is regenerative medicine? What unites something like this Zoominar, where we have the Department of Neurology and the Broad Stem Cell Research Center? And regenerative medicine is really one of the final frontiers in clinical neuroscience, in neurology, neurosurgery, and psychiatry. And that's because as the population ages, the brain is the limiting factor of aging. We have normal cognitive decline with aging. What was that person's name? Where did I put the car keys? But we also have major age-related diseases like stroke and dementia. And there are no therapies that deal with brain deterioration and brain disease. And so an interaction with the Department of Neurology and the Broad Stem Cell Research Center is really focused on regenerative medicine in the brain. How do we repair it? And how do we reverse or treat some of the decline that occurs with aging? So without further ado, we'll move on. Oh, and one other final note, if you have questions, that's great. Please submit them through the Q&A function at the bottom of the Zoom screen. And we'll have a session at the end for that. So we now move to our first talk, and this is from Dr. William Lowry. Dr. Lowery is a professor in the Department of Molecular, Cellular, and Developmental Biology. And he's had some really profound discoveries in basic stem cell biology and also to move these closer to the clinic in several different realms. And we'll hear a little bit of one aspect of his research, and that is as it relates to neuroscience and neurology. So thank you, Bill.
I'm Bill Lowery. I'm a professor in molecular cell and developmental biology at UCLA. I'm also a member of the Broad Center for Regenerative Medicine, the Johnson Cancer Center, Molecular Biology Institute. But really my primary affiliation has been with the Stem Cell Center because of my interest in using stem cells to model normal human development. So 14 years ago, we began working with human pluripotent stem cells. And the idea was to take these cells that can become anything and push them to become neural cell types or cell types of the brain. And we successfully were able to do this. We could make neurons and astrocytes, different cell types that make up the brain. And along the way, we figured out ways to purify them such that we could then look at individual cell types and understand what genes are on and what genes are off as cells go through a normal human development in the nervous system. And that led us to a number of discoveries about pathways that turn on or off. And that led us to think about ways to manipulate that cell fate. And we found that we could, by simply changing culture conditions, we could promote different cell fates and promote the maturation of different cell types. And it allowed us to model normal human neural development in a more sophisticated way. And it allowed us to think about ways to create very mature versions of human cells that we could study in vitro for the purpose of understanding genetic programs or programs where genes are on and off at different stages of development. And that then led us to think about, well, are there ways to study abnormal human development? And about 7 years ago or so, it became apparent that you could take a skin biopsy from any person and isolate cells from that, grow them in vitro in a cell culture dish. And then there's a reprogramming trick that we can use to reprogram those cells back to a very early stage of development and make human pluripotent stem cells from them. So we can now make human pluripotent stem cells from essentially any person using a skin biopsy. And we then started to do that process with people who have various neurological disorders, such that we could take people with, for instance, intellectual disability syndromes. So these are people who have low IQ, formerly considered mentally retarded is the old terminology for it. But we can take skin biopsies from people with these intellectual disability syndromes and reprogram them back to a pluripotent state, and then turn those cells into neurons and astrocytes, cell types of the brain, and then study the neurons and astrocytes that you create from these people with intellectual disability syndromes and compare them to the normal process. And in so doing, we've uncovered a number of different pathways that are abnormal in these intellectual disability syndrome neurons. And we're trying to use that information then to figure out ways to restore a proper function. So what we find when we take these intellectual disability syndrome neurons is that they are electrically active, as we know neurons are, but they don't communicate well with other neurons. The signals between the neurons get lost, and so you don't have the right balance between excitation and inhibition, and you end up with cells that look like they're essentially epileptic and firing too many electrical signals. But when we reverse the pathways that are misregulated in those intellectual disability syndrome neurons, we actually restore their normal function, and we can get those neurons to talk to each other effectively again. And so we're now thinking about ways to exploit this in terms of creating drugs that could do the same thing. We have identified one small molecule that can restore function in these intellectual disability syndrome neurons. And I think Ranmal Samarasinghe will describe this process a little bit in his talk. But essentially, we can restore function in these three-dimensional versions of these models of intellectual disability syndrome. And so taking that lead compound, we're now doing medicinal chemistry with folks in the chemistry department, and we're looking to leverage this with the TDG, the Technology Development Group, in order to raise more funds to exploit this possibility that we could create novel small molecule compounds that can restore function in these intellectual disability syndromes. And the exciting thing is that there are hundreds of these syndromes, and so we're talking about hundreds of thousands of people who could potentially benefit from these types of drugs. And because of our ability to take advantage of human pluripotent stem cells and the medicinal chemistry capacity here on campus, I think we're getting close to having the ability to develop potentially new therapies for people with these disorders, for which there's currently no therapeutics that can restore function or reverse disability in these patients. So it's also important to point out that this work that I described is the product of a number of really important collaborations that have been fostered by the Broad Center. For instance, I've worked extensively with Tom Carmichael to use neural stem cells as a model for repair in stroke and injury. And that's been an excellent endeavor where we took advantage of our ability to manipulate cell fate and create special types of neural progenitor cells or cells that will become neurons and astrocytes, and use those to repair stroke in a mouse model. We also worked extensively with Ben Novich in neurobiology and his postdoc Ranmal, and we used these interactions to exploit their ability to create 3D models of neural development, whereby they were able to take the cells that we had created from the intellectual disability syndrome patients and turn them into mini brains, or these organoid models that allow for a more exquisite interplay between neurons and to detect these neuronal connections and communication. And that allowed us to model intellectual disability syndromes in a much more sophisticated way. And it's also provided a platform by which we can screen small molecules for the ability to restore neuronal communication in these intellectual disability syndrome models. And we're excited about the infrastructure and the environment that has been created around the Broad Center for Regenerative Medicine, which has allowed us to work at the interface between developmental biology, human pluripotent stem cell biology, medicinal chemistry, and then technology development to allow us to potentially bring these methods to a translational or a clinical end. And I would applaud the Broad Center leadership for creating such an environment that allows us to do this work. And I look forward to seeing where it goes in the future.
Thank you, Bill. Dr. Lowery, that's great to hear. We have 3 short segments today, and Bill's is the first. We now segue to Dr. Ranmal Samarasinghe. Dr. Samarasinghe did his neurology training here at UCLA and his MD, PhD elsewhere, so he has a deep scientific background. And then during his fellowship, he did a really interesting thing that relied on some support from the Stem Cell Center's fellowship program. He wanted to understand how to apply regenerative medicine to the brain, and particularly how to understand how human brain circuits are involved in disease, to really directly model human circuits and not say those circuits in a mouse and then extrapolate. And so he has been a pioneer in the field of applying human cerebral organoids, which he'll describe, to the study of human neurologic disease. So thank you, Dr. Samarasinghe, and we'll go to your video next.
Good morning, my name is Ranmal Samarasinghe, and today I'm going to be talking about human brain organoids as a stem cell-based research tool for neuroscience. So, let me start this. I am an MD PhD neurologist and a new assistant professor in the Department of Neurology at UCLA, and I'm also a clinician scientist, and so I see patients with epilepsy and autism. The Broad Stem Cell Research Center has been really important in my research career. I did all the work that you're going to see today in Dr. Ben Novich's lab, and he's a center researcher. He's my postdoctoral mentor. We've had a close collaboration with Bill, who's speaking today. And I'm the recipient of Broad Stem Cell Research clinical fellowship that allowed me to actually complete my postdoctoral work, and has resulted in me starting my own lab recently, where I'm interested in using stem cell technology to understand brain disease, in particular epilepsy and autism, but hopefully with implications for other neurological disorders. So how do we hope to do this? So modeling the brain and physiology is complicated, And the point of this first image is just to show that the human brain is large, it's complex, it's got a lot of parts, and it's bigger than a lot of the models that we use to study brain in the laboratory, like mouse brain or monkey brain. The human brain is bigger, that is. The brain is also this physiologically active organ. And so what you're seeing here are actually brain waves from a patient, from something called an EEG, or an electroencephalogram. And EEG is obtained in this way, where we put, it's a cartoon showing electrodes placed on the scalp of a patient, and we can get this kind of a signal. And clinically, as an epileptologist and a clinical physiologist, this is something that we do routinely, and it provides information on brain health. But at a deeper level, what the EEG actually shows is a coordinated activity of brain cells, of neurons within the brain, that are firing in such a way as to be able to produce these brain waves. So the task at hand is to go from a stem cell and produce this model that that is able to reproduce this anatomical complexity that you see in human brain, as well as this brainwave complexity, right? This electrical complexity. So how could we possibly do this? So our approach is to really go from this really big, complicated organ, complicated problem, and try and simplify it. And so I'm showing illustrations here of a brain and of like a picture of a circuit, of a computer circuit, essentially, that looks complicated and has a lot of parts. And the idea is to take a small piece of this, of each of these, as an example, and then extrapolate from that small piece into the larger problem. And that really is fundamentally how I think of you going from stem cells and using brain organoids as a tool. So what is a brain organoid? So it's derived from a stem cell, including from a human stem cell of a patient that has a particular disease. And this is an image of a brain organoid. It is this three-dimensional structure that's grown in a laboratory dish, and that's brain-like. So why is it brain-like? Because in this cartoon, what you're seeing is it has components that are seen in actual human brain, components that are actually not seen, sometimes seen in other brain, like rodent brain. And it has this really nice anatomical organization. This is a microscope image of an organoid, and the different colors represent different cells that are separated, just like you would see an actual brain. Now, we knew all this when I started my research in Ben's lab in the BSCRC. But what we were more interested in knowing is, does this also result in sophisticated EEG-like activity in brain waves? And so to do this, of course, we have to come up with some new tools to use with organoids. And we have two major tools that we use. And one is using a sophisticated microscope called a multiphoton microscope to look at the actual activity of individual neurons within an organoid. So what you're seeing here is a brain organoid derived from a stem cell, and the flashes you're seeing is individual cells lighting up during a recording. And then we've developed tools to actually look at that data and pick up the individual cell firing patterns. The other tool that we use, other than this multiphoton calcium indicator imaging, is kind of like an EEG for an organoid. It's called a local field potential recording. What you're seeing here is a picture of the actual process. So we make these electrodes and we stick it directly into the organoid, and we can get an image, or excuse me, data like this. And so what you're seeing here, if you can see my cursor, are these peaks and valleys. And each of these peaks actually represent the actual brain waves from the organoid. And remarkably, these brain waves actually look like what we'd see in an actual human patient. So we can reproduce some of that physiological complexity in human brain in the organoid. And so just to recap briefly, we can make organoids from a patient's own stem cells. It has a sophisticated anatomy. And what we've really been working on to push this technology even further is to measure brain waves in the organoid. And we saw very early on that these brain waves are actually similar in the organoids to what are seen in actual human brain. Okay, so how are we applying this? So for our first studies, we actually had Bill's help, because Bill had stem cells from a patient that has a disorder that's associated with autism and epilepsy. It's called Rett syndrome. What you're seeing here is the calcium indicator imaging that I described earlier from a normal organoid. So this is a brain that doesn't have any disease. And you're seeing, hopefully in the video, individual cells firing one after the other. And below is a plot where every yellow kind of streak represents an individual cell. In contrast, when we made an organoid from a patient with the Rett syndrome, with this genetic disease, it looks different. And I hope you can appreciate this in the video, but what you're seeing actually is individual cells firing, as well as this kind of the entire organoid firing together in these flashes. And I've shown that below in the plot where we have the yellow streak kind of all going together. And that's what's highlighted in the red dotted boxes. And these flashes are actually like what are seen sometimes in patient EEG. It's very similar to an epileptic-like state. And in other words, we can go from the patient stem cells to the organoid and recreate some of the pathology that we'd expect from physiological measurements in an actual patient. And of course, I said we're using 2 techniques. So what about the other technique I described, this organoid EEG approach? So here's a normal, but to describe the normal, let me show you the patient. So this is a Rett patient again in red. And at the top, what you're seeing is the raw trace, what we call when I first stick the electrode into the organoid, this is what I see. And the raw trace in the normal is this kind of smooth crescendo decrescendo figure, right, in blue. In the patient, it looks really different. We're seeing all these lines sticking out, and these are called spikes. It's highly abnormal. Then when we go to look at the actual EEG part of it, where we want to see these peaks and valleys, we see that in the normal. None of that in the case of the Rett syndrome organoid. It's just like a flat descending line. And so what we learned from this is that we can reproduce this epileptic-like state in the organoid from a patient that should have epilepsy. And we use this to learn more about why this is. And I won't get into that here in the interest of time. Instead, I'll talk about another important potential use of organoids and that we used, that we tested with Rett syndrome. And that's in application in drug discovery. So as I mentioned, Rett syndrome is associated with epilepsy. Epilepsy is a disease without a medical cure. The best you can do for a patient with epilepsy is give them drugs that they have to then take for the rest of their lives. That's the best you can do. And so epilepsy, by the way, is defined by recurrent spontaneous seizures. And unfortunately, for 1 out of 3 patients, about 30% of our patients don't even respond to the newest drugs out there. So despite the best drug treatment available, they still have seizures. And this hasn't improved in over 100 years of trying. And so what does this suggest to us? We need new models to try and understand epilepsy and to treat the disease and to come up with new drugs. And maybe organoids are one of these models. So here is the Rett EEG recording, EEG-like recording from a Rett organoid without any drugs. And so you see a lot of these spikes like that I showed before, and you don't see brain waves. Then we tried to add a traditional anti-seizure medication. In this particular case, we actually saw fewer spikes, which is good, but we didn't see any brain waves coming up. So that's not good. Then we tried a novel drug called pifithrine alpha. Once again, this is from Bill and his own studies in Rett, where he found that this drug can be effective. And we saw a reduction in spikes, just like with the traditional drug. But what was really great is we actually also saw some restoration of brain waves, suggesting that in this model, using this novel drug, we can get more of a rescue than we've seen before using other models and using other drugs that are traditionally used to treat Rett syndrome, at least epilepsy and Rett syndrome. So in conclusion, essentially what we were able to do is use the model that we have with organoids in this particular disease to, first of all, see abnormality, find out why that abnormality is happening, and to test some new drugs. So where are we going with this? One idea is personalized medicine. And so what do I mean by that? Well, this is an example of an EEG, and it's really abnormal. It's from a patient that has a really, really devastating genetic epilepsy that basically is present at birth. At around 4 months is when this happens, and it has no cure. Here is our calcium indicator recording from organoid with the same genetic mutation, right? So you can see the similarity, but these are different patients. The idea, sort of the goal in the long term, is to go from a single patient, get stem cells from that patient, make organoids from that patient, and then, for example, test drugs so that you can have personalized therapy for that patient, or maybe even personalized understanding of why that patient has that disease. So that's one sort of future direction. The other idea is I don't want these organoids just to be used for what I'm interested in, which are epilepsy and autism, for example, but broadly used to study brain disease. What I didn't mention earlier when I said we're making brain organoids, specifically, we can actually make brain organoids for very specific parts of the brain. including this part of the brain called the hippocampus. And the hippocampus is where human memories, or where memories generally for any animal, where our memories are formed. And so we did make hippocampal brain organoids, and what we discovered is that these hippocampal organoids have brain waves that are very similar to what is actually made in real hippocampus, in an animal or in a human. And these brain waves are associated with memory formation and memory consolidation. And so this is really remarkable, and as many of you know, there are a lot of diseases where memory is affected. And so we've been working with some of our collaborators that work in these disease areas, like Dr. Chao Peng in Parkinson's disease and Dr. Jess Rexek in Alzheimer's. And the goal now, for example, here is to make organoids that have pathology associated with memory loss. And then we can see what happens to these brain waves. Are they gone? Why are they gone if they aren't? And if they don't go, why is that? So really understand disease mechanism. So in summary, what I really wanted to illustrate overall in terms of brain organoids and their utility is the way that I think about an organoid. And that is as this tool to really unlock the awesome potential of a stem cell, to go from this simple yet very highly potentiated thing, the stem cell, to something that we can then use to study even more sophisticated problems. And really, in my mind, nothing gets more sophisticated than the brain. So we can use the organoid now to study brain anatomy and also brain physiology. And with that, hopefully, go forward in enhancing our understanding of neurological disease processes and come up with new therapies that we haven't in the past. So inclusion, thank you. This has been really collaborative work throughout UCLA, as well as from collaborators outside of UCLA. I've had a lot of funding sources that have been important. Certainly the Broad Stem Cell Research Center has been one of those. Thank you.
Thank you, Dr. Samarasinghe. That was very interesting, and really kind of highlights how even basic science and stem cell biology can be applied to studies that lead to investigations of diseases, in this case, epilepsy, and used as a screening tool for possible novel drug development in that field. So the final talk is from my lab. This will also be fairly quick and to the point. This will be live. We thought we'd mix it up, some recorded and some live. In addition to the leadership positions in neurology and the Broad Stem Cell Research Center, my own lab is very interested in the ways in which the brain can be stimulated to repair after stroke and in dementia. And we have had really important collaborative activity with Dr. Bill Lowery, who you've heard from, and he anticipated this a little bit. And I'll describe 3 projects that relate to regenerative medicine in the brain, stimulating brain repair. And I'll describe them kind of in descending order, with the most clinically relevant and the most further developed towards the clinic, and then some of those that are closer to discovery of these 3. So one of our most important projects relates to a finding that we had back in 2010, and that was that stroke damages a particular area of the brain, and we know that. But the interesting thing is we found that it reduces the ability of the brain to signal in areas around it that are not directly damaged. So stroke alters the intact brain, the surviving brain, and diminishes its function. And it does so in the areas that are supposed to recover, the areas adjacent to the stroke. To back up a minute, we recognize that there are over 7 million people in the US who live with a stroke, and there are 800,000 strokes a year. And the most significant risk factor for stroke is age. And so this incidence and this prevalence will go up with time. So here's a disease with no therapy for repair and with this devastating burden. And what we found is that the brain is inhibited in its recovery because it reduces its signaling ability, stroke does, in the areas adjacent to it. We further then identified some of the molecular signaling molecules that are affected by stroke that reduce this signaling. And this is interesting because what it shows is the brain stroke lowers the excitability of the brain adjacent to it. It can't get excited in its normal state like it would before stroke and fire new signals. And so if you can't move your forearm or you can't individuate the fingers of your hand after a stroke, the adjacent tissue, which should normally recover and take over that function, can't, because it can't signal as well. So we identified 2 signaling systems that contribute to that low excitability. And these are in a neurotransmitter system called GABA, and a second inflammatory signaling system, a chemokine system called CCR5. We identified drugs that block those abnormal signaling systems. They enhance recovery in mice with stroke, and we've moved to clinical trial now with a large consortium in the US and Canada, and big pharmaceutical companies have taken up the GABA story and have initiated clinical trials in Europe and in Australia. So this is a finding that really is core in regenerative medicine and biology, because it looks at how the brain decays after stroke, unexpectedly finding that it can't signal as well, even in the tissue that looks normal, that's adjacent to stroke, and leads to new molecules that might reverse that process. A second study in the lab relates to stem cell transplantation in stroke, pardon me. And this is done in really close collaboration with Dr. Bill Lowery. And so one of the nice things about UCLA as a scientist and faculty member is that it's the largest UC campus by people on the smallest by land size. And so the stem cell biologists, the clinical scientists and the clinicians are all really together. And so Bill and I have interacted by going to each other's seminars and running to each other in the hallway. And Dr. Lowery had found in his lab, as he alluded to, that he could bias basic stem cells, so-called iPS cells, to become glial cells with a simple small molecule therapy. And this is a big deal in the field because previous protocols required months of iterative and slow kind of nudging of stem cells to become these kinds of glial cells. So Bill's technology could do this in actually several days. And we recognize that this would have an opportunity to treat a particularly devastating form of dementia, so-called vascular dementia. This is the kind of dementia in which little strokes accumulate in the brain, in the connecting areas of the brain. The ancient anatomists called this the white matter because that's how it looks in autopsy. So the white matter carries connections between hemispheres in the brain or between areas of the same hemisphere. And even in normal aging, even if we are healthy, exercise, we don't have high blood pressure, at 80, all of us start to accumulate little teeny strokes in this white matter. And then in vascular dementia, this is massively increased. And then a further damning fact here is that vascular dementia accelerates Alzheimer's, and Alzheimer's disease accelerates vascular dementia. And the most common kind of dementia is actually mixed dementia, the two occurring together. And so we started to study whether this new stem cell therapy could promote brain repair in vascular dementia in a new mouse model that we developed specifically to be more accurate in modeling vascular dementia. And we found that this new stem cell therapy, which is an iPS therapy that differentiates into a glial cell, does indeed repair the brain in mice models of dementia. It enhances the formation of new connections, and it enhances the circuits that are damaged in white matter, these areas in which there's a so-called myelin sheath. It remyelinates these areas. We've now moved on with direct funding from the California Institute of Regenerative Medicine and NIH, and developed assays and other studies that are necessary to take this scientific lab discovery and move it to a human cell product so that it could be an off-the-shelf or out-of-the-freezer approach to repairing the brain in vascular dementia. So a final project, there are many in the lab, but a final project that I thought would be interesting to discuss is enhancing the brain's normal repair processes. I think when all of us think about stroke, in friends or family members, we know that some people get substantially better, many people get partially better. There is a normal but limited process of recovery in stroke. And so what is that process, and can we remove the brakes? And my lab has found that stroke stimulates a growth factor response in the brain. And interestingly, this growth factor response sort of looks like normal brain development. So when the brain normally develops, it has to form connections and it has to strengthen those connections. And after stroke, the brain essentially can become, in a term, sort of new again. It can reactivate some of these growth factor programs that are normally present in development. But in the adult brain after stroke, the problem is it just doesn't do a good job. It's very limited. So I have identified what growth factors these are and some of the reasons of why this is limited. So future studies will be to enhance that process through specific drug therapies to promote recovery. So that's a kind of a snapshot of what's going on in my lab. And I alluded to a maiden function that enhances the success and kind of runs as a theme throughout Dr. Lowery and Dr. Samarasinghe in my lab. And that is the collaborative interactions at UCLA that are fairly unique. And this is because of the physical structure in the post-COVID era, soon approaching, we'll resume this kind of tight physical interaction. And it also relates to a culture of collaboration that's really bottom up. Collaboration doesn't work when it has to come from the top. Collaborate, say the leaders. It comes really from a natural sense of interaction that occur in the faculties, as I mentioned, in the hallways and in the symposia. And it leads to a lot of energy and a lot of scientific discovery. So I'll conclude with that. We'll now move to address some of the question and answers that have come in. I can also interpret some of those question and answers and ask direct questions. So a first question that came in related to seizure disorders. And the question asks, has there been any luck or progress in understanding seizure disorders or developing new therapies? And of course, Dr. Samarasinghe squarely has that in his clinical and his scientific interests. So I'll let him field that question.
Sure. Thank you for that question. So yeah, I hope you were able to see some of the videos that we showed earlier and some of the data that we've generated in our lab together with Bill and Ben. And the answer to the question is yes. The problem with seizure disorders, and maybe what you're referring to, is the fact that we've had some limitation in the efficacy of the drugs that are available currently to treat seizures. And what we have shown already with this very preliminary work with using pifedrin alpha, which is the therapeutic that Bill Lowery, Dr. Lowery has first discovered, is we can see some increased gain in terms of our ability to not only prevent the actual kind of hyperactivity of the cells, but also maybe rescue the function of the circuit. So another way of putting that is yes, I think the organoid model has shown us that theoretically, and certainly from some data that we already have, that there is some potential for moving forward in terms of new therapies. The problem is that some of the mouse models that we've been using so far have kind of come to this roadblock where we can't advance more and we're stuck in terms of the efficacy of medications. So yeah, I think that there's certainly more room to move forward, but the important point, I think, is that we're able to model these circuits and we're able to model the human circuit using these stem cell derived organoids. And so potentially in the future, we may be able to come up with even more candidates.
Great, thank you, Dr. Samarasinghe. Second question relates to CRISPR technology. And the question is, how can this help with the kinds of studies that we've described here, but also in the field of regenerative medicine in general? And Dr. Lowery, this is something you think about a lot. It's probably a good one to pass off to you.
Sure. So it's pretty exciting. For those that don't know, this CRISPR-Cas technology allows you with relative ease to manipulate the genome of cells in culture, and perhaps if you can deliver it in a human setting, there are some clinical trials on the way, you could correct genetic aberrancies that lead to particular diseases. And so people are very excited about this technology. It just won the Nobel Prize this last year. And people were thinking about ways to apply it to different diseases. In the immediate term, it is enormously useful for bringing disease modeling in vitro because you can induce particular mutations, deletions, or duplications that lead to disease, and you can do that in a patient's cells in the lab quite simply, and then look to see the way my lab and Ranmal's lab have shown that you can model diseases in vitro. You can recreate those diseases quite easily by doing this genome manipulation in existing cell lines. But longer term, yes, there's a lot of excitement around building on gene therapy work, which is the ability to introduce genetic material into a tissue or into an entire person to add something that's missing in disease. But CRISPR builds on that and allows you to take something away or make a specific change to the DNA that's in the genome of the cells that make up the tissue that people are walking around with. And so it's quite exciting. People are thinking about it for things like muscular dystrophy, where you have a defect in this gigantic protein that regulates strength in your muscles. And what they've been doing is using this CRISPR-Cas to correct the specific genetic mutation and create a more functional protein to restore function. This is all still in the preclinical phase, but there's lots of exciting efforts for a wide variety of genetic diseases specifically because of the ease with which you can manipulate the genome.
Thanks, Bill. Yeah, it's really been a revolutionary element, primarily because of that final statement. It's not hard to do this. It's hard to get it into a clinical therapy, we recognize, for many of the reasons we've discussed, but most labs can now access this technology, at least for basic biological study, to turn genes on and off. A third question is, Dr. Carmichael mentioned a therapy his lab discovered is being tested in clinical trials in Australia and Europe. What is happening with this in the US? That's a great question. So this is the CCR5 story. This is a chemokine. The molecule's name is CCR5. And we discovered, well, actually almost 2 years ago now, that stroke unexpectedly turns this CCR5 molecule on in brain cells and neurons adjacent to the stroke site. And this inhibits recovery. It diminishes excitability, that first story I mentioned. And the interesting thing here is that CCR5, we discovered it in stroke and limiting recovery, but it has a much better job, much better known job, and that is it's the co-receptor for the AIDS virus. And so there's a deep pharmacology already present to blocking this receptor. So we were able to piggyback on that. With the novel finding of its role in stroke, we could use a drug that 15% of AIDS patients are on and apply that in a therapy to see if it would work in stroke recovery. And so we've started that clinical trial. We're working with Yale, Burke Rehab Institute in upstate New York, and UCLA. And we've recently brought in a large coalition in Canada. So we're just getting off the ground with that. And that will be a clinical trial for stroke recovery. Looks like other questions are coming in, which is great. One question that we received before that I was going to get to and is now restated, thank you, is regenerative medicine applicable for trigeminal neuralgia? Why is there not a cure for it? And I can feel that one. Trigeminal neuralgia is actually a syndrome, as the person who wrote this may recognize. It's a syndrome because it's distinguished by the hallmark of pain in the face, rather than by its cause. There are many causes that can induce the pain syndrome. Some of them relate to abnormal blood vessels that may be too close to the nerve cell bodies. Some of them may be because the nerve cell bodies are too sensitive and light touch or other things set them off. And so there is no single therapy that will work, but there is movement forward to understand if there is an enhanced sensitivity in trigeminal neuralgia. So I would expect that clinical neuroscience in general will soon close around ways in which we can diminish the sensitivity of trigeminal neuralgia and diminish its disease impact. I'm sorry, there isn't a direct answer that, yes, here's this new pathway that may lead to it, but there's a lot of research in this area. Okay, next question is, what impact could your discoveries have on the development of future treatments for Alzheimer's disease? So this is a good question, I think, for all of us. I'll start. So Alzheimer's disease can be tackled in a couple of different ways. One way we're approaching it, I mentioned, and that is Alzheimer's disease almost never exists alone. It's a part of a mixed dementia. The abnormal proteins that accumulate in Alzheimer's disease occur in the setting of these little white matter strokes, and the two accelerate each other. So Dr. Lowery and I have a really promising stem cell therapy to repair the brain in that situation. Another possible approach is to develop new and more relevant human circuits to study Alzheimer's disease and its pathology. And Dr. Lowery has really taken a lead in this and is in a good position to answer that end of this issue.
Yeah, we've been studying syndromes that are like Alzheimer's in terms of premature aging and thinking about how cells under stress respond to stress through essentially protecting themselves and becoming dysfunctional as neurons. And so we're looking at ways of relieving that stress in vitro models of neurons, and with Ranmal and Ben Novich, and thinking about doing this in organoids. So we're thinking about doing this in vitro, really, so that we can look at cells from lots of different related syndromes, lots of people from different genetic backgrounds. And then maybe Ranmal can speak to work going on in the Novich lab, thinking about building more sophisticated three-dimensional models that allow you to, seed tau tangles or amyloid plaques or the kinds of pathological features, and so recreate Alzheimer's in vitro, and then use the kinds of assays that Ranmal showed you for function and dysfunction, and then it allows you to think about different ways of treatment and to try out those treatments in an in vitro setting in the dish in the lab where there's no risk to the patient.
Thanks, Bill. I can just tack on right where Bill ended. And so what Ben and Bill and I have been working on from the organoid perspective is, of course, trying to build this system where we can model a circuit. And I talked about circuit in the context of epilepsy, but this certainly applies to Alzheimer's disease as well. And that's sort of what I tried to end with in my last slide. And so as Bill said, what we've been working on now is to have these organoids. So now we have a platform where we can see what a normal circuit looks like. And when I say circuit, I mean interactions between healthy neurons or brain cells and how they function, what their physiology is. And we've been working with collaborators within the Department of Neurology to essentially seed these organoids with hallmarks of Alzheimer's disease. And this is of course just work that's starting out now, but the idea is then use the same exact assays that I've already developed to look at what happens to the circuits, what happens in terms of the functionality of the organoid. And of course, we can also section the organoid and look at the anatomy of it, how does it change in response to this. And as Bill mentioned, we can use the same approach and test a lot of therapeutics. We can grow 100 organoids in one batch and test all kinds of different drugs, for example. So I think there are a lot of avenues to build on the fundamental platform, which is the stem cell-based organoid to look at Alzheimer's disease.
Yeah, in a sort of a lay summary of that work, how do you give an organoid Alzheimer's disease? And why would you want to? And you'd want to because it's a human brain circuit. And so it has a certain amount of ground truth to it. It's not a mouse that you somehow put a human gene into. And so Dr. Samarasinghe and Dr. Ben Novich, who we've mentioned, who's really a critical collaborator here, have worked to try to develop a way in which you can give an Alzheimer's disease syndrome to an organoid and then really study its basic biology. And it hasn't been simple. As a matter of fact, the field in general has failed here. But working with David Eisenberg at UCLA and others, there are possible ways forward using a couple of different parallel approaches that really could herald a cutting edge way to understand human brain circuits with Alzheimer's disease and do things like Dr. Samarasinghe mentioned, screen drugs. And to go back to that first question, a Crispr screen is one way to do that. There are a variety of ways to move more rapidly once this kind of brain organoid platform is in place. We have another question that relates to Parkinson's disease. How has this work advanced Parkinson's disease research and what studies will soon be launched? Parkinson's disease was thought to be low-hanging fruit in regenerative medicine because we know there's largely one cell type that's lost that accounts for a lot of the symptoms. We know where it occurs in the brain. And it was thought, well, we'll just plunk stem cells in and they'll change into that cell that's lost, and voila, you have a therapy. That was about 20 years ago. And we've realized that that really doesn't work. You could cause really severe side effects if the cells go the wrong direction and become something else. That happened in several clinical trials. But the most common problem has been the cells just sort of sit there. They don't differentiate. They don't really do anything. And so the field is focused back further into the lab. How do you make these cells so that if they go into the brain of a human, they understand they're supposed to become, in this case, a dopaminergic neuron? And that field has now progressed over about 10 or 15 years, and there are several therapies that look more promising. I still think we're 7 to 10 years out, but there are some clinical trials starting in that area, and they're not actually advanced yet, so they're not entering patients. But I think in the next 4 or 5 years we'll see some of the first trials, but it may take 5 to 7 before these show anything. Another question is, what can we learn from intellectual disabilities regarding aging? Somehow, can studying intellectual disabilities help researchers understand brain aging? Dr. Lowery?
Yeah, you took the words right out of my mouth. So we have noticed, and one of the primary features we see when we study intellectual disability syndrome neurons in the lab is that it was always thought to be due to a failure to develop appropriately. It was thought that these things strike in utero or just after birth. And so, you know, the neurons are just failing to fully mature and develop. What we're finding is actually very different, that they make an attempt to mature and develop and send out all the appropriate connections that neurons do. But they, due to the mutations that cause the disease, they actually undergo a stress response. And it's a stress response that is typical of what you would see in very aged neurons. So if you look at neurons in a very elderly person, or in neurons that come from an aging type syndrome. So we're actually thinking that these intellectual disabilities are really caused by premature aging. And so that's how we got into this stress response issue, and we start looking at drugs that relieve stress response, and that's really anti-aging type methods. So all the types of pathways that turn on when a cell is in trouble due to aging or genomic abnormalities, etc., those stress responses actually instruct the neuron to retract its dendrites, or the little outgrowths that it uses to talk to other neurons. But if we can relieve that stress, trick the neurons into thinking they're not stressed or everything's okay, they go on and they send out their connections, and they form appropriate connections and form neural networks, as Ranmal has been able to detect with the brainwave studies. And so we are thinking about these ID syndromes, intellectual disability syndromes, as premature aging. And another interesting one is Down syndrome, where I think everyone's pretty familiar with Down syndrome, and the kids that grow up with this and the types of symptoms that you see is very common with the disease. The issue is that these kids also prematurely age. They have a very early onset for Alzheimer's in their 30s and 40s. And in fact, that's typically what causes these kids to pass is premature aging type syndromes like Alzheimer's. So we're now getting cells from kids with Down syndrome and culturing them in vitro, as we see this as an authentic bona fide premature aging syndrome. And we're trying to apply our anti-aging strategies to these to see if these could work in Down syndrome patients. And at the same time, they provide a very interesting model for the study of Alzheimer's disease. We suspect that these cells that we get from the Down syndrome kids are going to be more susceptible to getting Alzheimer's disease even in the dish. And so that's part of this work around trying to seed organoids or cell cultures with these plaques amyloid or tau tangles and things to see if the Down syndrome cells are more susceptible. And if so, we could try to employ our anti-aging strategies in these syndromes that affect very young people. So it's a sort of a different way of thinking about aging, but we're excited about the possibility because there's lots of known aging pathways that we can target.
Thanks, Dr. Lowery. We're running out of our allotted time slot, and we have several really great questions. And so we'll respond to these directly. And feel free to send more in. The one thing about this is it's such a new field, regenerative medicine in the brain. And UCLA happens to be so good at it. We have a Department of Neurology focused on it, a Broad Stem Cell Research Center focused on it. We run the largest neuro rehab hospital in the state, Cal Rehab. So we have a clinical, basic, and applied scientific infrastructure. So it's a pleasure to talk about this and to provide the kind of the expert responses to questions that you send in. So we have a time for a few final remarks. Dr. Lowery, did you have anything to close with?
Well, I'd just like to thank again the Broad Center for creating a platform for collaboration. You know, when I came to UCLA 15 years ago now, I was actually a hair follicle biologist, and I still do that. I still do study hair follicle stem cells. But being surrounded by all these interesting scientists studying human pluripotent stem cells and neural stem cells and models of neural diseases, I quickly got wrapped up in it, and now here I am studying intellectual disability syndromes. And that couldn't have happened. I couldn't have gotten pulled into that direction where we're now making drugs that potentially could alleviate intellectual disability syndromes were it not for the Broad Center and all the environment that it fosters of collaboration and bringing scientists both physically and intellectually together to ask important questions and reduce barriers to getting interesting novel projects started.
Great, thank you. For some reason, I find the hair follicle research resonating. Dr. Samarasinghe, do you have anything to finish with?
Sure. Thanks, Dr. Carmichael. So I'll just say, as a trainee that's just transitioning into my independent position, there's two things that I'd mention about UCLA that really stand out. One, in terms of the science you've seen today and the other science that goes on at the center, the absolute adherence to the highest scientific standards and rigor. As a trainee, this is something that I appreciate, and we're going to be bringing forward into my own lab. You know, this is not necessarily the case everywhere. Certainly I can say that it is the case at UCLA. And just to reiterate, as a final point, something that Dr. Carmichael has already brought up, and Dr. Lowery as well, but the level of interaction between clinicians and scientists. So the medical center is just behind me, just across the street. And some of us actually go and work in the clinic, and we can bring what we see in the clinic back to the lab directly. But even if that's not the case, this is a very collegial community, and one in which there are a lot of interactions, not just between clinicians and scientists, but between different realm of scientists. And this is unique, and I think it's led to some unique opportunities for collaboration and advancement. And it's something that I've already experienced in a way that I hadn't before coming to UCLA as both a trainee and now as a new faculty member.
Thank you, and this concludes our show. I want to thank all the participants again and all the people that contribute to and interact with the Department of Neurology and the Broad Stem Cell Research Center so much. As you get a flavor for here, most of the truly innovative sort of blue sky directions that we take occur because of generous support. They occur rapidly, quickly, in a nimble intellectual environment that's really enabled by engagement with our philanthropic supporters. So thank you again for your support. Please send additional questions in, and otherwise have a great morning.